Beta oxidation. Beta oxidation is the biochemical pathway that hydrocarbons such as lipids are processed for the generation of energy. The products of beta oxidation are FADH2 and NADH, as well as acetyl-CoA. The acetyl-CoA will then be used in the Krebs cycle for the generation of additional FADH2 and NADH that will be metabolized in the electron transport system, as well as ATP. Beta oxidation begins with the fatty acid in the form of an acetyl-CoA. The transformation begins with the second and third carbons from the CoA. The enzyme acetyl-CoA dehydrogenase that used FADH to make FADH2. This will place a double bond between the two carbons. Next, water will be added to the double bond, and this will place a hydroxyl group on the third carbon. Next, the enzyme hydroxyacetyl-CoA dehydrogenase will use NAD to remove a hydrogen, forming NADH, and a double bond will form with the oxygen in the hydroxyl group. The enzyme beta-ketothiolase will place a thio-CoA between the two double bonded oxygens on the chain. This will cause acetyl-CoA to be released from the remaining acyl-CoA molecule. In this manner, two carbons are removed from the fatty acid with each cycle. The products are FADH2, NADH, and an acetyl-CoA. The remaining acyl-CoA will repeat the cycle until the last four carbons are released as two molecules of acetyl-CoA. The energy that is produced comes from the products that either enter the Krebs cycle or the electron transport chain. When we think of fatty acids, we are constantly being told about the benefits of unsaturated fatty acids. These come in many configurations, but all have double bonds somewhere in the middle. The process of beta oxidation deals with the standard saturated fatty acids, but needs modification with the unsaturated ones. Therefore, there are changes with the part of beta oxidation when double bonds are reached. The initial steps until the double bonds are reached are those that we just showed with the release of acetyl-CoA. When the double bond is reached, an enzyme 3,2-trans-enoyl-CoA isomerase will change the orientation of the double bond so that the beta oxidation can occur. However, this rotates the remainder of the molecule. There is no real problem if there are no additional double bonds. Otherwise, the fatty acid would need to be modified to change the position of the remaining double bonds by using the enzyme 2,4-dienyl-CoA reductase and NADPH and a proton to reduce the two double bonds to one. At this point, the remaining double bond is between carbons 3 and 4. This then gets shifted to between carbons 2 and 3 by the enzyme 3,2-trans-enoyl-CoA isomerase. At this point, the rest of the fatty acid can be degraded using standard beta oxidation. While most fatty acids are even numbers, occasionally there will be an odd number fatty acid. With these, the process continues until it reaches the remaining 3-carbon hydrocarbons. This 3-carbon molecule is propionyl-CoA, which is then carboxylated to form succinyl-CoA. The succinyl-CoA can enter the Krebs cycle directly, but does not result in additional energy being produced. Beta oxidation is the process that is used to break down straight-chain hydrocarbons and harvest the energy or the acetyl-CoA building blocks from them.